welcome back. We'll continue on with sex determination and sex chromosomes. Let's find out some ways that other organisms determine sex and a little bit about why we can get along with a bunch of X's and still function as if we don't have too many chromosomes. Okay. The answer to that is bar bodies. So bar bodies are when you take one of the X's, of the two X's, or the how many X's you have, anything beyond one X is wound up so tight that that information inside of it is inaccessible. Bar is named after the person who discovered them. So this doesn't refer to the shape or the structure of the chromosome. It's just Bar discovered them and called them after himself. Bar bodies are in anyone that has more than one X, whether it's XX, XXX, or XXY, or any of the other combos we talked about. During early development, one of the X's, hard to say, one of the X's is inaccessible. It's wound up tight and made inaccessible. So this person who has XX, in each one of their cells, either bar bodies this one or this one and really has one functioning X. This person in each one of their cells, because they have more than one X, actually bar bodies everything but one X. This person bar bodies one X per cell and ends up with an XY. It is random which X's get bar bodied. There's a series of really interesting experiments that show this. It highlights it in your text if that's your thing. I'm not too worried about you repeating that experiment to me. We know that the inactivation of one of the X's or all the extra X's are random, and all but one is deactivated. And so this begs another question that you're probably sitting here thinking, wait a minute. Because I told you that people with Turner syndrome, where you have only one X, or people with Klinefelter syndrome, where you have two X's and a Y, have symptoms from the fact that they have too few or too many X's. And if one X is always active and all the other ones are deactivated in all humans, why would these matter? Well, two parts to that. About 15% of the extra X's escape deactivation. So in some of your cells, if you have two X's, you are expressing both X's. And those X's are deactivated and bar-bodied after initial parts of development. It's very early, but it's not at the zygote stage. It's later. That means that they do seem to play a role in early development that can lead to some consequences in the adult phenotype or in the born phenotype. I want to show you what it looks like. The lion hypothesis, the part of the support for it is calico cats. I have a video in the little page that I have for this part 7 and chapter 7 and 8 lecture that's called Why Women Are Stripy, that explains this based on human development. What happens in calico cats is color in cats is on the X chromosome. So this cat has orange and black. That's a B. These are the colors that are on the X. But one X is deactivated in every cat's cell. I can't explain the white patch on the face with this explanation, but I can with others. Sort of like dog color coat genetics, it's complicated. This cat is orange and black as well. And you see the stripes? Stripe, stripe, stripe. What you're actually seeing is the cell lines as that cat developed. During early development, this cat had two X chromosomes. One X carried black and one X carried orange. Early development, before it was obviously cat, it shut down one random X in each. And then as those cells went through mitosis, they passed on a open copy of the X and a bar body. The same, during mitosis, the same X stays bar bodied as it's passed from cell to cell. That means that as the cells develop inside the cat and become coat color, the cells that were active and using black these were all probably from the same cell line that grew through mitosis and developed the skin here. The cat's skin here developed from a different cell line and became orange. Same here, you can see these stripes. These stripes are actually, you can actually see this as an expression of how the cat actually developed in patterns. These are expressions of cells that are direct descendants of each other, expressing color as part of their role as cells in the body. This can result in mosaics, in where you have a pattern of expression where you see blotches or colors, but also an expression of disease where I see a partial expression in part of the body of a disease and not in a disease in another part of the body. For here, I'll do it here. These look like they have antenna now. For things like being able to see color or hemophilia in humans, we talked about in that practice day in lab when we practice questions that those are also on the X chromosome. Hemophilia is the clotting is carried on the X chromosome for hemophilia A. So if you have a bad allele and you only have one X, you express hemophilia, which is an inability to clot. Color blindness, the inability to see red and green, is on the X chromosome, meaning that the ability to see red and green is also on the X chromosome. When you inherit from your mom a good color vision gene, and from your dad a bad color vision gene, the person that grows up with this two X's can see red and green. They have an allele that allows them to develop the cones in their eyes that see red and green. But in some of their eye cells, they're using this X. In some of their eye cells, they're using this X. And actually, when we do the colorblind test, people that are carriers for colorblindness will see those colors differently. My father was colorblind. When I take the colorblindness test at the eye doctor, I actually fail one part of it, and it always causes the woman flipping the thing really fast because she's not expecting me to be colorblind. She always freezes and then goes back and then freezes, but that one is actually the one that is testing for, like, you just don't see colors quite as well as everyone else. It is a measurable part of the phenotype that you are carrying it. It's just some parts of my eyes are expressing my father's X, which had color blindness on it. Some parts of my eyes are expressing my mother's X, which did not have color blindness. I know because my brother can see color. The X inactivation center is involved in deactivating the X, or bar bodying it. So this mechanism that we create these bar bodies is called imprinting. Imprinting is a weird word. Like, I always associate it with like pushing into something. But imprinting is when part of a chromosome or a different chromosome makes a substance that blocks the expression of a different gene. On the X chromosome, there is an X inactivation center. And we think that the X that expresses this X inactivation center protein is actually the one that gets bar bodied. So this is interesting, and bear with me. We will talk about it later as we talk more about imprinting. The X inactivation center on the X chromosome contains the gene XIST. It stands for X inactivation specific transcript. All this codes for, it's not a protein. All it codes for is a really long piece of RNA. So it gets transcribed, but that RNA does not code for a protein. Its name is, and see what I mean about just memorizing the letters. Its name is X inactivation specific transcript, really long, non-coding RNA, or just ink RNA. Don't call it that. Call it X inactivation specific transcript, long, non-coding RNA. When this is made, it attaches to a different protein that starts coding the X that is making this really long piece of RNA. that X gets coded in the protein, and that causes it to be unusable. It gets imprinted. And so when it's imprinted, it gets kind of, I don't know, I always imagine it like being wrapped up in plastic so it's inaccessible. That X now cannot be used. This is carbon copy. This is the first cat to ever be cloned. And this is her surrogate mom. Her surrogate mom was, the egg was implanted in her, but carbon copy doesn't share surrogate's mom DNA. When carbon copy made kittens, do you think they looked exactly like carbon copy, knowing that cat color is on the X chromosome? No, they did not. They did not look exactly like carbon copy, even though they were copies of carbon copy. Carbon copy also does not look like the cat she was cloned from, because the X chromosome deactivation of the bar bodies is random. So I've lied to you a little bit, not in a harmful way, but I've lied to you a little bit to make the fruit fly examples in the earlier classes easier. Kind of lied to you a little bit about other things too, but I'll build on them without, because I didn't want to overwhelm you with genetics. I guess we'll just make things a little bit more complicated now, but now you have the background to get it, right? Fruit flies totally have an X and Y chromosome, and male fruit flies are XY, and female fruit flies are XX. But the Y in fruit flies does not make them male. They have them, and they have code on the Y, and it's less code than on the X, but there's no gene for testes on the Y in fruit flies. Sex in fruit flies is actually determined by the ratio of autosomes to sex chromosomes. And I've got this map here, this chart here, because the ratio can be kind of bonkers. If you have 3 sex chromosomes for every 2 autosomes, we'll get something called a meta female, which actually doesn't develop. She'll actually have a lot of X's inside of her gametes, if she could make them. If the ratio is 1 to 1, so for every autosome there is an X, she'll develop as female. If the ratio of X's to autosome is 1 to 2, the flies develop as male. I'll put the rest of it here. If the flies have a ratio of sex chromosomes to 3 autosomes, they'll form a meta male, which has some other interesting chromosomal abnormalities. Depending on the ratio, female, male, meta, and meta, we can also develop flies that are intersex. That means that they'll develop both testes and ovaries, or characteristics of both male and female fruit flies. And this is probably only happening reasonably in the lab. During development in wild fruit flies, it's unlikely that we would have surviving adults from anything from but 1 to 1 and 1 to 2 ratios. Maleness is on the autosomes that every fruit fly has. It's only activated in the absence of the second X. So weird. This is about ratio of sex chromosomes to autosomes, not about a gene that then suppresses female development. This is C. elegans. It's a nematode that's been really well studied. They are hermaphrodites. Hermaphrodites comes from Hermes and Aphrodite. It means that they have both male and female ability to make gametes. A hermaphrodite makes egg and sperm. They generally do not self-fertilize. That you might as well mitosis if you're doing that. It's a lot of extra wasted energy, but they can. In the case where they cannot find something to mate with the same species, they can self-fertilize, use their own sperm to fertilize their own eggs, and that will produce almost all hermaphrodites who will be younger and able to do their own things. These are going to be clones, but a little bit less than 1% will be only male. That means they can only make sperm. When I breed a hermaphrodite and a male, I'll get a 50-50 ratio of hermaphrodites to males. There are no strict females in C. elegans. You're either a hermaphrodite that makes both sperm and egg, or just a sperm maker. Cross-fertilization results in 50-50 ratio. Self-fertilization results in a 99-1 ratio. I have a segue for you. And oddly, that segue, I love that word. It doesn't look like it's written the way it says. Bees, honeybees, do not have sex chromosomes. Diploid bees are female. Haploid bees are male. So if you have 2 sets of chromosome, you develop as female. If you have 1 set of chromosomes, you develop as male. This leads to some interesting relationships in the bees, and is probably the source of them being eusocial or truly social insects. Honeybees are social in a way that most other insects are not. If you kind of play this out, and this is a totally different class to play this out in, but so just trust me on this. If you play out their heredity, the female bees that are worker bees inside the hive are actually 2/3 related to each other. So they are more related to each other than other siblings because their dad only gave them 1 set of chromosomes. Their mom, they actually got a set of chromosomes from their mom and their dad, whereas the male bees only get 1 set. So there's some other things at play here. When a female queen bee mates, she can actually, she takes in lots of different male sperm. She has a container inside of her that stores all of that sperm. And we now know that she can pick and choose which male is, even though he's been dead for 5 years, she can pick and choose which male sperm she uses. So often when we look at the worker bees in a hive, they're all full siblings at each season. So they're very related to each other. Darwin really questioned in his writings how honeybees could sacrifice themselves the way they do, guard the hive, live the kind of lives they do in a way that made sense with natural selection and evolution. Haplodiploidy and the weird relationships they have because of it, genetic relationships, actually explains the evolutionary drive to sacrifice yourself for the hive. Because you actually are really much more related to your siblings than humans are related to their siblings. And you're kind of saving your own genetics by saving the hive. So slightly different class, but if you're interested in this, I will talk about it till you can't stand it anymore. I just wanted to note it in there. They have ploidy as their sex determining system. Things like turtles, a lot of reptiles that are cold-blooded, and a lot of amphibians use temperature-dependent sex determination. I actually have a whole segment in my environmental science class about this because it's really become an issue with global climate change. As the temperature has changed in the beach, we're skewing the sex ratio of turtles in some cases to where they all hatch out as females. And that's a problem because if there's no males, we don't get any more new turtles. This is very common in reptiles. I'd worked at a research project when I was in undergrad where we were rearing turtles to try and save them, and we didn't know this. So we reared them all at a uniform temperature, and they all hatched out as the same sex. Hundreds of turtles, but no ability for genetic diversity. In the nest, when they lay their eggs in a big pile and bury it. The ones that are at the bottom of the nest stay warmer and they develop into females. The ones at the top develop into males. They're cooler. And again, I can talk about this some more too, if it's your thing, or I can give you the data for turtle sex determination for the green turtles, which actually we've had a measure of success in restoring after discovering this sex determination system. So a little bit of practice with me before I go off the rails and stuff that I'm super interested in, and you probably aren't. Maybe are. Some practice about bar bodies. This is what bar bodies really look like. This one here, here is a bar body, so you can see it. It gets wound up tight and imprinted and then bound to the inside of the nucleus. You can see it with a microscope, and it's a really easy way to determine sex. It's caused some problems if people are not the expected XXXY. where you identify if you were XXY, you would also see bar bodies like this. If you stained skin cells and saw a bar body, what do you expect to see? And then what do you expect to see in a man? So someone who is expressing the Y chromosome and yet you see bar bodies. So this means that he has two X's and a Y. It does not mean if you have two extra Y's, if you're fiddling around between these two, Y's do not get bar bodied. If you have extra Y's, they would both express. And this is a possible genotype for people. When we see this, we generally just see a little bit higher of testosterone production. Bar bodies are only going to form here and here. But because he has a Y, I think C is the answer. I know C is the answer. You observed the cells and karyotype of a novel species, that means it's brand new, of mammals found on an island. The females do not have a bar body. They all have two X chromosomes. Males have one X and one Y. Considering dosage compensation. Did I skip a slide? No. Let me just, in case I didn't, it's been a long day. Considering the fact that you must have one X, and that each individual must have the dosage of genes from one X, what do you predict? So females don't have a bar body. They all have two X chromosomes. The male is XY. How do I make sure that each one of these has the same number of genes they need to live their lives? So he's got to have the same dose of X as she has. I don't want to re-record the whole video, but if just in case I did skip a slide with dosage compensation, this is basically you need everyone in order to be human is going to need a certain amount of that protein. If XX is what you need, he needs to have XX worth of genes, even though he has only one X. He's going to have to transcribe his X at twice the rate of the female. He'll get twice as many proteins out of his X, and she'll use each X just once for every time he uses an X twice. XIST RNA plays a crucial role in X inactivation. This is the thing that generated the really long sequence of RNA. The really long sequence of RNA coats the X chromosome that produced it, and whichever X has more of this XIST coding it, it becomes the inactive X. If a mutation inactivated this gene that makes the really long RNA on only one chromosome, what would happen? Inactivate it. I'm giving you a second to read it. It can't be deactivated. The mutant chromosome would always be active. I would actually think in this case, because the other X chromosome was making it, we would always inactivate the same X in this individual. So if they were a cat, they would only be able to be one color. Question to think of for when you come to lecture. Can male cats be calicos? And how? With that, I will finish topic number 7. I will see you in lab. Ask me questions. This is where I don't know. I think fun is something different than you do, but I think these are kind of fun to think about. Now we're going to have cascades of interactions. This is where we start to actually get into the real organisms expressing real things. I think this is where we can start to like touch and feel the genetics. And I will see you the week of, this should be the week we start the P-Glow experiments and a little bit about micropipetting. So once you do this, I'll do a lab with you about how to use the pipettes. And after that, it's very procedural in lab. So questions, or just have fun. All right, I'll see you soon. Bye-bye.